So we're looking at the immune system, and your first section of notes talked about the non-specific immune system. And that is going to be really important both for humans and the whole group of mammals, but also for other invertebrates. We do want to know that animals and plants have both been found to have complex immune abilities. And so plants are, when we look at animals, we have vertebrates and invertebrates, and so your invertebrates are going to have mostly your non-specific immune. So when we look at invertebrates, these are things like worms, insects, anything without an internal backbone, they're going to be all non-specific. They're going to be able to recognize that they, I did not spell, specific. They're going to be able to recognize that they've been invaded and protect themselves, but they're not going to be able to recognize if they're being attacked by the same thing over and over again. And so when we start to talk today about the specific immune system, the specific immune system is able to use cell communication and specific receptors to identify antigens or invaders and protect against them for the future as well. Now plants, this picture here, remember, is an example of a plant, and it is going to be able to show you that plants are able to do the same thing. They're able to recognize when they've been invaded. One of the key things that I want to mention about plants is that plants are often able to sacrifice other cells to contain, so they will sacrifice cells to contain an infection. So you can basically do an experiment with any plant in that if you damage the leaf and you come back a day or two later, you'll see that either a larger section of the leaf is dead or sometimes the whole leaf falls off. It'd kind of be like the human body saying, oh, I have a paper cut and an infection on my finger, I'll just kill the whole finger and regrow the finger. That isn't how it works for animals, but plants are able to recognize when they're infected, try to target that infection, and include sacrificing cells that could prevent further infection. So our main focus today is actually going to be on how the immune system helps maintain homeostasis and how cell communication or interaction between these cells plays a really big role in that. If you go back and look at your curricular framework, you see that there's a whole section on how a variety of defenses affect dynamic homeostasis. So having a variety of ways to defend your body, your organism, is a way to maintain homeostasis in a changing environment. Looking at the rest of the curriculum, we're also looking at the idea that we have cell communication and cell signaling. So we're focusing on how the immune system can maintain homeostasis and how cell signaling is a big part of that. All of this hinges on the idea of an antigen. Now, an antigen can be an entire invading organism, or it can be part of the organism. What happens with the antigen is that you're going to have a set of cells that are going to be able to identify that that doesn't belong. And so we see here this idea of B cells being activated and C cells being activated. And so this idea of activated is based on identifying, not self. And so what does self mean? Well, what self means is that all of your cells have what's called an MHC. And that's called a major histocompatibility complex. And your MHC, your major histocompatibility complex, is a collection of surface proteins. Every single one of your cells has a collection of surface proteins that identify your cells as self. And so your B cells and T cells, which we'll get to their jobs in just a moment, your B cells and T cells are constantly monitoring the body. They're exploring all parts of the body, and they're literally bumping into things. And their surface proteins should match your surface proteins. And as long as their surface proteins are matching your surface proteins, when the B cells and T cells are circulating, they're going, oh, self, 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 self. But when they run into something that does not have your major histocompatibility complex on it, it goes not self. And this is an antigen, and the antigen will trigger an immune response. So by either the T cells or the B cells running into the antigen, or by the macrophage actually presenting it. Remember, macrophages are nonspecific. They just engulf it and destroy it. The B cells and the T cells are then able to begin their cascade. If you look at this picture, you can very clearly see there are a series of events that are going to happen. We're going to talk on the next page about what exactly B cells do and what exactly T cells do. But the key here is this idea of the shapes, the surface proteins. All of your cells have certain proteins or shapes, and your B cells and T cells are able to identify them as self. This antigen can be a microorganism. It can even be parts of a microorganism. It can be things like pollen, which isn't a living thing. That's the sperm from a plant in a pollen grain. And so lots of things can act as antigens and trigger these immune responses. What we're going to talk about on the next page is exactly what B cells and T cells do, but the key idea being that we're able to respond especially to dynamic homeostasis or changes in the environment. So let's look a little bit closer at what exactly B cells and T cells do. T cells are what are part of what's called your cell-mediated immunity. And these names don't necessarily directly tell you anything. These names are from long historical research, and so they're not necessarily up to date and very informative. But I do want you to connect the T in T cells to the T in mediated. That will help you there. And then I want you to think of them as your fighter cells. And so I connect all of those T's. T cells, cell-mediated, and fighter cells. These are your cells that are going to attack the specific antigen. Now you have these other cells. You have these macrophages and these natural killer cells that are able to attack antigens as well. But we're focusing on attacking a specific antigen, which means that your T cell needs to know what it's going for. So it's not just running into things and going, not self, I destroy you. It's actually going, oh, I know you, and I know exactly how to destroy you. So when we look at T cells, we do have to note that they are mobilized. They have to be activated. And a big part of that is going to be what are called cytokines. Cytokines are chemical messengers. And so if you were to refer back to the previous picture, you see that these cells are all interacting with each other. It's kind of like coordinating an attack. That's exactly what the specific immune system is able to do. It's able to look for and search out specific antigens. And so again, it all has to do with the antigen, and we see here these antigen fragments, meaning we're going after a very specific antigen. And the T cells are actually able to attack and kill infected cells, and they're really able to attack and kill these antigens. And so there's a lot even in this picture that you don't need to know. The key about T cells is it's called cell-mediated immunity, and they are the fighter cells that attack the specific antigen when signaled. So the key is that these cells are only going to be activated when they receive the correct chemical messengers, and then they're going out to try to destroy the antigen. The T cells are a really big cycle of turning on other T cells. What we're going to focus most on are B cells. B cells are part of what's called your humoral immunity. And B cells, the way that I remember what they do, is they produce the antibodies, B cell antibodies. Antibodies are proteins to tag the antigen. Be very careful. The antibody does not destroy the antigen. It's just a tag. It's like a tracking device. And so the antibody has to tag the antigen. Well, the tag needs to be specifically shaped. Right now in your body, there are cells, B cells, that are constantly making antibodies. And so these B cells, when they run into an antigen, will actually display the fragments and then begin to actually mature into antibody-producing cells. So what we see with the B cell is that it will actually engulf the antigen as well. But then it says, oh, I now know how to attack this. And so it makes these tracking devices. Now we need to talk a little bit more about these antibodies for just a moment. When you see this antibody, there's something on the antibody called a variable region. And the key about the variable region is that we're able to make slight changes in the amino acid sequence equals a large variety of shapes. And so if you remember from our protein unit and our protein synthesis unit, the way that we're able to do this is because of our introns and our exons and what's called alternative splicing. So there are a certain number of antibody genes, but then the way that they are spliced, introns and exons, in and out, we're able to make a huge variety of different proteins. And so that's the advantage of the antibody, is we're able to make thousands of different shapes. Now, one of the keys is that these antibodies will be produced in small amounts forever once activated. So once your body recognizes and is able to design an attack for that antigen, your body will continue to produce those antibodies for really long periods of time, actually for forever. So that means that the next time you're invaded, so our very last key point is about your immune response. Is going to be quicker the second time. Because of this specific immunity, knowing how to attack that, how to track it, how to contain that immune invasion, your response is actually quicker the second time because you already have these cells designed for that antigen. Unless the antigen is a different shape. So the advantage of your specific immune system is that you are able to recognize what something is that invaded you and how to attack it and how to get rid of it, specifically through these tags, antibodies. And as long as that's what you're being invaded by, you can attack it quicker and you usually don't get sick.